The phospholipid bilayer is a selectively permeable double layer of phospholipids. Selectively permeable means that the cell membrane controls what is let in or out of the cell. Phospholipids are made of two fatty acids, a glycerol, a phosphate group, and a polar molecule. The phospholipid tails or fatty acid tails are on the inside of the phospholipid bilayer, attached to the negatively charged phospholipid head. The heads are hydrophilic, which means that they are attracted to water, while the fatty acid tail is hydrophobic and repels water. Together, the head and tail create an amphipathic molecule, which is a molecule with hydrophilic and hydrophobic parts. The cholesterol is also on the cell membrane and is inside and helps to work with cell signaling and cell communication. Within the cell membrane are integral and peripheral proteins. Integral proteins are embedded in the bilayer where hydrophobic residue interacts with the fatty acids and binds them to the membrane. Peripheral proteins are membrane proteins that temporarily attach to the membrane that they are associated with. They attach to integral proteins or penetrate the peripheral regions of the phospholipid bilayer.
The hormone receptors bond with hormones, and normally only one specific hormone receptor binds to one type of hormone. When a hormone binds to a hormone receptor, this stimulates protein creation through transcription and transcribing. The interaction between hormones and the receptors determine cell growth in various areas of the body and other stimulation. Hormone bonding sites are both integral and peripheral. An example of a hormone is insulin, and the hormone receptor is tyrosine kinase. An enzyme that is attached to an insoluble material is known as an immobilized enzyme. Immobilized enzymes can provide increased resistance to changes in conditions such as pH or temperature, which connects to the body's need to maintain homeostasis. It also allows for enzymes to be held in place during the reaction and separated after for repeated use. An example would be penicillin G acylase, which is an immobilized enzyme. Immobilized enzymes are both integral and peripheral. Cell adhesion is a type of structure that exists in tissues in multicellular organisms that consist of multiprotein complexes that provide contact between neighboring cells and spaces. They help to enable communication and reduce stress on cells. They are particularly abundant in epithelial tissues. Cell adhesion structures are both integral and peripheral. Cell-to-cell communication is achieved through the secretion of proteins, small peptides, amino acids, nucleotides, steroids, dissolved gases, and more that bind to the receptors. Cell-to-cell communication is both integral and peripheral. Channel proteins transport water or certain ions by forming a protein-linked passageway across the membrane. It does not require energy and allows for water to move freely into the cell. Channel proteins are integral. Pumps for active transport are used to transfer macromolecules across the cellular membrane. Some examples of the pumps are P-type ATPase, which is the sodium-potassium pump, calcium pump, and proton pump. F-ATPase is the mitochondrial ATP synthase and chloroplast ATP synthase. V-ATPase is the vacuolar ATPase. ABC transporter is composed of the MDR and CFTR. All of the pumps are integral.
We have our original drawing starting with the hydrophilic heads and the hydrophobic tails. This is an example of an integral protein in the cell membrane. We then have a carbohydrate chain, protein pump, this is an example of a hormone and a hormone receptor, which can be either integral or peripheral to the cell membrane. This is another example of an integral protein. And a peripheral protein and a protein channel. Protein channels are all integral to the cell membrane.